Hello everyone, I'm Riikka Lund. I work here in the Turku Bioscience Centre as a head of Finnish Functional Genomics Centre and also as a group leader of Biomedical Epigenomics group. Today I will give you a short presentation of our recent projects where we have assessed potential of cell-free nucleic acids as biomarkers for diseases. Please join me for the presentation. So as many of you know, our group, Biomedical Epigenomics Group, operates in close connection with the Finnish Functional Genomics Centre, and we support the activities of the core facility by implementing new methods for genome-wide research, and we also use these methods to reveal new knowledge how genome is regulated during early development, in aging and in complex diseases. And today I will be focusing on our two recent projects on cognitive decline and Alzheimer's disease. And why it's important to study Alzheimer's disease is that Alzheimer's disease is the most common cause of dementia worldwide. And it's also among the most common causes of death in aged people. There is a strong genetic component. Based on twin studies, approximately 50%, there is over 50% heritability. And then also the environmental and lifestyle factors influence the disease risk. And this problem is acute because the number of patients with Alzheimer's disease is increasing rapidly due to aging of populations. And this is creating a major burden for our healthcare system and economy. And at the moment, we don't have any early diagnostic markers, and there are also no disease-modifying therapies available. And in order to develop better more efficient disease-modifying therapies, we would need these early markers which would enable early intervention to the disease before too much neuronal damage has taken place. So to address this problem, the goal of our study was to identify novel markers for decreased memory performance, which is early sign of Alzheimer's disease. And we wanted to identify these markers in peripheral blood that would enable efficient, high-throughput screening of individuals. And later on, we would then validate these markers as prognostic markers for Alzheimer's disease, as these patients will be followed up also in the future. So the detailed aims of our presentation were to implement and evaluate performance of two methods to detect cell-free circulating nucleic acids, both DNA fragments that are methylated and also cell-free microRNAs circulating in the plasma. And then the second aim was to find out whether we could find markers for decreased episodic memory performance. So what is the rationale behind studying these cell-free nucleic acids is that these nucleic acids can be DNA fragments or microRNAs or RNAs that are released from tissues, and they can be detected in body fluids like urea or plasma, and they could be used to detect diseases like cancer or monitor disease status or drug responsiveness, and they are already routinely utilized in clinic in non-invasive prenatal testing to detect chromosomal abnormalities in fetus by using mother's blood. And when we started this study, or planning this study in 2013, this method was very new, and there were not many studies that would have used this method by using genome-wide analysis. So we were using, we had lots of open questions also remaining whether this method is suitable for the purpose. So first we wanted to see whether we could detect these circulating DNA methylation marks associated with episodic memory performance in the plasma. And why we wanted to look into the DNA methylation is that the DNA methylation signatures are tissue-specific, and we could assume that there would be cell-free fragments released from the degenerating neurons from brain that are circulating in the body fluids, and these were kind of the fragments we were tracking for. And the other cause which could cause such marks in the cell-free fragments is that DNA methylation is also sensitive to environmental influences, and this disease process itself can cause such changes in the DNA. So that's another alternative why we would detect such markers in the cell-free DNA fraction. So this study was carried out in collaboration with Biocenter, and it was started in 2014, and during 3 years they interviewed over 1,100 twins to identify twin pairs who would be discordant for memory performance. And based on the phone interviews, they identified 18 twin pairs that were discordant, and these twin pairs were invited for more thorough clinical characterization, including PET imaging and neuropsychological testing. Based on the clinical characterization, unfortunately, there were even less discordant pairs, but we anyway collected blood samples from all of them. And from those blood samples, we fractionated the plasma, collected the plasma, and isolated cell-free DNAs from the plasma from 7 twin pairs that were available at that time. And from that cell-free DNA, we then proceeded to the bisulphate conversion. So when we carry out the bisulphate conversion, we will convert all the cytosines that are not methylated to uracils, and all the cytosines which are methylated remain intact. And then by reading these fragments, we can distinguish which of the cytosines are methylated and which are not, and to what extent at the scale from 0 to 100%. From these fragments, we prepared libraries for next generation sequencing, and then we read the content of the fragments with Illumina HiSeq sequencers, and then we carried out data analysis to recognize what fragments were present in the plasma and what was the DNA methylation level. However, before we could identify the marks that are associated with episodic memory performance, we had many open questions related to the sample preparation, as the method was new, so we didn't really know how it works. So we had questions like which anticoagulant is good for the sample preparation, how much plasma is needed for the sample preparation, is it possible at all to measure DNA methylation in the cell-free DNA fraction, and how deep sequencing do we need for this purpose? So to answer these questions, we first run pilot analysis where we compared 2 different anticoagulants, including EDTA and citrate, and we didn't see any significant differences in the anticoagulants. Cell-free DNA was successfully isolated from both. So we selected citrate for the continuation, as in some cases it provided better yields of cell-free DNA. Then next we compared the plasma volumes. So we had 500 microliters, 700 microliters, and 1 milliliter. And when using 500 microliters, we noticed that 2 of the 3 test samples failed, but then with higher amounts, all the samples were successful. So we concluded that volumes 700 microliters and more are safe to use for this purpose. Then we also examined whether the amount of the cell-free DNA will influence on the number of CPC sites that we can detect in the genome. And perhaps surprisingly, we didn't see any impact of the starting amount of DNA on the genomic coverage. Any amount between 200 nanogram and 80 nanogram worked well. Then we also examined whether the sequencing depth will influence on the number of CPC sites we can detect in the genome. And again, with the sequencing depth we were using, we didn't see any impact. So we could conclude that the sequencing depth we were using was sufficient. And this means that the sequencing depth that, for example, if we have 10x coverage, it means that we will be reading each CPC sites of the genome about 10 times. However, then when we looked into the number of CPC sites we detected, it varied from 4,000 sites to 13,000 sites, which was quite low in each individual, as we know that there are about 28 million CPC sites in the genome. So it looks like there is rather small proportion of the genomic DNA present in the plasma that can be analyzed. And this creates especially the problem when we then wanted to compare the twin pairs that were discordant for the memory performance. So we had at this point 4 discordant twin pairs, and when we took all the CPC sites that were detected in all of the twin pairs, we had only 2,500 CPC sites left, so it's very small proportion of the genomic CPC sites. However, we continued to compare the differences between the twin pairs and found no statistically significant differences, which was perhaps not so surprising as there were so few sites to study. It's perhaps worth mentioning that there were also some limitations related to the study. The bioinformatic method was not fully optimal, as at the time the method that was available didn't allow us to compare the continuous variable, which would be a memory performance score at in this study. And we were forced to compare the groups based on clinical diagnosis, those individuals who were cognitively preserved in comparison to those who had decreased memory performance. Also, our sample size was small, so the statistical power was weak. However, these pilot results were not convincing enough for us to continue to more individuals. So we concluded that this method is perhaps not so optimal for this purpose. But as a conclusion, we can say that the DNA methylation can be measured from cell-free fractions. However, the proportion of the CPC sites we can detect is rather small. We can also conclude that the plasma volume starting from 750 microliters are robust for the sample preparation. We can conclude that both anticoagulants EDTA and citrate can be used. However, in this study we were not able to identify DNA methylation marks for episodic memory performance, and we concluded that the study is perhaps more suitable to study one individual, for example, patients with cancer, and then you could ask whether there is DNA fragments released from the, for example, tumor to the plasma, and you could monitor the disease status and even try to track the origin of cancer. But in this kind of situation where you want to compare a group of individuals, this method is not so good, I would say. Okay, then in the other part of the study, we then asked whether we can detect cell-free circulating microRNAs associated with episodic memory performance in the plasma. And the rationale was the same. Also, microRNA signatures can be tissue-specific, and we could assume that there are microRNAs circulating in plasma which are released from the dying neurons. And there could be also microRNAs which are specific to the disease process that could be detectable. So in the beginning, again, we compared two different isolation methods, this time commercial kits from QIAGEN and Exicon, and the plasma amounts that were used were 50 microliters, 100 microliters, 200 microliters, and 400 microliters. And we could conclude that both kits gave sufficient amount of cell-free microRNAs. However, we then at the end choose the QIAGEN kit because the profiles looked perhaps a little bit better than with the Mercury kit, but both kits could have been used for this purpose. Then the study design was the same, the samples were the same, but this time we now had one more additional twin pair, so we had altogether 8 twin pairs in the analysis, and we started isolation of the microRNAs from 400 microliters of plasma. The RNA yields varied quite much between individuals, so we obtained about half a nanogram to 3 nanograms of RNA from each individual. We prepared the libraries from half a nanogram of cell-free microRNAs, again did the next generation sequencing and data analysis. Again, we looked whether the sequencing depth will affect the number of microRNAs that are detected in plasma. And again, we didn't see any major impact of the sequencing depth. So by using read amounts from 500,000 to 2.5 million, we could detect kind of the same amount of microRNAs. There was more variation between individuals rather than caused by the sequencing depth. So quite small sequencing depths are good for this purpose. Then we looked what types of small RNAs we could detect in each sample, and as expected, we could see lots of reads mapping to the microRNAs. In addition, we could detect lots of reads mapping to the miscellaneous RNAs, transfer RNAs, also exogenous ribosomal RNAs with non-human origin, mainly bacteria and Candida albicans. We could also detect reads mapping to mitochondrial transfer RNAs. And then there are these 3 samples which also had high number of reads mapping to protein coding genes. And this is something that is not wanted, and it may indicate that there has been contamination by the peripheral blood mononuclear cells in the plasma fraction. However, when we looked more carefully into the profiles and number of microRNAs that were detected, we could see that these 3 samples were still good to go, but then there was this one sample which had very high number of protein coding RNAs, and that was clearly affected. Both the number of microRNAs was lower, and also the abundances of the microRNAs were lower. So based on that data, this sample was excluded from the analysis. Then interestingly, there was also this other sample which had only 17,000 reads, although for most of the samples we had more than 500,000 reads. And interesting finding was that also with this low amount of reads, we were still able to detect 50 microRNAs in this sample, which was quite good number if you look into the number of microRNAs that were detected in other samples with up to 6.5 million reads. So this is another plot illustrating the microRNA abundances between different samples after normalization. And here is the sample with high contamination of protein coding RNAs, and it was excluded from the study as it has clearly affected the abundance of the microRNAs detected in the samples. Then there was this one sample where we had low number of reads, but after analyzing the whole data set with and without this sample, we concluded that the statistical models were more stable when this sample was included, and the low sequencing depth didn't affect the results too much. We just lost the low abundant microRNAs for that sample. So it was included in the final analysis. So we then compared the individuals who were discordant for the episodic memory performance. And as a reference, in the MIR base, there is at the moment a total of 1,917 microRNAs that are annotated. And of these microRNAs, we were able to detect 336 microRNAs in at least one individual without filtering out the low signals. But after removing the low abundant signals, we had left 45 microRNAs that were detected in at least 5 cases. And we didn't require that these microRNAs would be detected also in the healthy individuals, as they could as well be absent there, as they are perhaps not having problems with the memory impairment. However, when we looked into the data, almost all the RNAs we detected in all the individuals, including the cognitively preserved ones. And then we run the statistical analysis with linear mixed effects model, and based on the results, there were 9 microRNAs that were associated with episodic memory performance in these individuals. And here are 2 examples. microRNA 10A and 10B are different isoforms of the same microRNA. And based on our results, depending on the model, they were also influenced by ApoE genotype, which is associated with Alzheimer's risk. They were also influenced by age, psychosity, and gender. But interestingly, based on previous studies, both are already also known markers for Alzheimer's disease. So microRNA 10A, for example, has been detected in individuals with Alzheimer's disease, and it has been found to modulate synapse function in animal models. And microRNA 10B has previously found to be early marker for Alzheimer's disease in the cell-free microRNA fraction in plasma. So very promising results, consistent with the earlier studies, and indicates to us that this method was working well, and we can actually detect microRNAs in plasma associated with the memory performance. Then if you look these plots, in the first plot we have the individuals grouped based on the clinical diagnosis. So we have the cognitively preserved ones and then those which have impaired memory, and there is a clear difference. And then in the second plot, we have the individuals plotted based on the episodic memory performance score and the microRNA level. And you can see that there is also quite much variation between the healthy individuals, so they have different memory performance score, and that correlates with the microRNA levels. And then in the cases, it's even more pronounced. And here in both cases, there is this one case which is behaving differently, and that one case belongs to the different genotype group ApoE44, which is the ApoE group that has the highest risk for Alzheimer's disease. And also based on our earlier study on whole blood, we could see that the ApoE genotype strongly influence these profiles, but unfortunately here we had only this one individual, so we couldn't control this specific genotype properly, but it looks like that's also influencing perhaps the microRNA levels. So as a conclusion, we were able to detect plasma cell-free circulating microRNAs with next generation sequencing, and the method seems to work very well. We successfully analyzed the profiles from as low as half a nanocron for cell-free RNA, which is really good because many of our key researchers are also working with samples from small children, and then you don't have much samples to use. So this method is probably very good for that purpose as well. Caution is needed when you fractionate the plasma to avoid the contamination by the peripheral blood mononuclear cells. And we could also conclude that we can achieve very good coverage of microRNAs already with rather low sequencing depth, which is of course good because the sequencing is really expensive. We could also identify microRNAs associated with the episodic memory performance. The limitations of our study are that the statistical power was not optimal. However, this study material from clinically characterized twins is really unique and challenging to obtain, so the results are still really valuable and encouraging. So here is our research team and collaborators who were involved in this study. We are very grateful for all the contributions and also to the core facility for the support, and we are also grateful to all our funding foundations and sources of funding who have made this study possible. Thank you.